Hey folks, hope you're doing well. Most of the research that I've done with cover crops over the past 25 years has been with winter cover crops. But 2 years ago, I started working on fall-terminated cover crops. And I did this in collaboration with 2 Ph.D. students from Stanford University. The video that you're going to see here is based on a presentation that they were invited to give to the agricultural expert panel, which is part of the Irrigated Lands Regulatory Program. So I took the recording of their presentation from last December, I edited it a little bit, and that's what you'll see in this video. Hope you find this interesting. Feel free to let us know if you have any questions or comments. And thanks for your interest in our research. Take care.
Thank you so much for letting us talk today. My name is Sierra Castaneda, and I'm here with Anna Gomes, my colleague. We're 2 graduate students from Stanford University. And today we're going to explore how non-legume fall-terminated cover crops might reduce nitrate nitrogen leaching and some potential implications to consider for the ag order. So we're excited to share some data that we've been working on the past few years with Brennan here as well. And we're going to start with an overview of the nitrogen cycle, just to get everyone on the same page, then explain the timeline of a fall and a winter terminated cover crop system in the Salinas Valley, then share the results from both a laboratory incubation and a field trial that we did, and we'll finish with some key takeaways to discuss. So just to start, I am going to define some key three processes that are really critical to the nitrogen cycle when talking about cover cropping. First, nitrogen mineralization is the conversion of organic nitrogen into ammonium, with immobilization being the reverse process of this, so going back from a plant available form into organic nitrogen. Secondly, since there's plenty of oxygen in most agricultural soils, another group of soil microorganisms convert the ammonium into nitrate, and this is called nitrification. Third, since the soil is negatively charged and nitrate is also negatively charged, it doesn't stick on the soil and is washed down the profile, either with irrigation or rainfall, and this is called nitrate leaching. So just keep these 3 things in mind as we're going about the presentation. So I'll say these a lot. So let's start with an overview of how we think about non-leguminous cover crops in the context of the Salinas Valley and the Central Coast as a whole. First, say a grower harvests their final cash crop of the season. This could be a broccoli or a lettuce, and the residues of that crop, along with the excess soil nitrogen, are left on the field. Then, prior to the rains in the winter, let's say a farmer plants a non-legume cover crop. This could be a rye or a triticale, 2 common cover crops in the valley. And so as that cover crop grows, it's going to scavenge or take up excess nitrogen from the soil in their roots and biomass and tying it up in the biomass of the plant tissue. And then once these cover crops are terminated, the residues are then incorporated into the soil to decompose, and the nitrogen mineralizes back into a plant available form. And so ideally, this process happens such that the next season cash crop, so the lettuce that's planted in the spring, can use this quote unquote recycled nitrogen from the cover crop residues, preventing it from being lost from the system and maybe even reducing the next season nitrogen inputs. So now I'm just going to briefly describe 3 possible management timelines to show when cover crops are used in the Central Coast. So first, which is the most common system used today, is a no cover crop or fallow system. This is kind of the business as usual. So after the final crop of the season is harvested, the field is then put into beds and left bare over the winter until the next season cash crop is planted the following spring. However, this leaves the soil bare over the main rainy season, and since there are no roots to take up excess nitrogen, the risk of mineralization, nitrification, and losses via nitrogen leaching is extremely high. So here's a more ideal system where a cover crop would be planted prior to the winter rains, usually around October, and grows over the winter, accumulating nitrogen in its biomass. And so decades ago, Dr. Louise Jackson from the UC Davis lab showed that winter cover crops can reduce nitrate leaching by up to 70%. However, this system can be difficult to manage because termination of winter cover crops can really complicate field preparation for farmers, and the planting of late winter and early spring crops could be delayed. So based on interest from growers, we explore here a third system, which is growing a non-legume cover crop over the fall period for a shorter period of time, around 60 days. And perhaps these fall-terminated cover crops, in addition to winter cover crops, could also help to reduce nitrate leaching. We're going to explore how this might lead to an opportunity for fall-terminated cover crops to get a partial credit in the ag order.
So this leads us to our main question guiding our research. Can fall non-legume cover crops help protect the groundwater by reducing nitrate leaching? Let's first start with our laboratory incubation. So this plot is from a 112-day laboratory incubation we conducted in the lab with rye residue from 4 different carbon to nitrogen ratios. So you can see that on the x-axis from 10:1 to 30:1. And the y-axis is the percent of that residue nitrogen that has mineralized by day 112. And across 3 different temperatures that are all relevant to the Salinas Valley. So we measured how quickly the residue mineralizes or releases its nitrogen from organic to inorganic nitrogen. And we found that as the carbon to nitrogen ratio increases, the percentage of residue nitrogen mineralizes decreases, as you can see the large arrow on the plot. So in other words, as a cover crop matures and gets tougher or more carbon rich, less juicy, as Eric would like to say, there's less nitrogen that's released in 112 days. And you can see this QR code here on the screen leads you to the paper that we just got accepted in the Journal of Environmental Quality. And we're also working on a YouTube video for this paper. So the second plot that we'll show from the incubation, we just pulled out 2 of the residues that we studied, the 19:1 and 30:1, and that's the range that we found in our field trial as well. The red dashed line, horizontal line that you see here in the plot is actually the threshold between net nitrogen mineralization and immobilization. And the highest percentage nitrogen we found for the 2 residues were 20% or 40% of the residue nitrogen that was measured in the warmest conditions of 20 degrees Celsius. In the plot here, you can see the red arrow that the net mineralization of the 30:1 residue only happened at the warmer temperature. So the higher the temperature, the more mineralization happening. So while these are really insightful and incubations in general are insightful to think about dynamics of mineralization, we want to think about in the field how this might change. And in the incubation, you can see this picture on the bottom left. The residue is undisturbed for 112 days. And we know that's very different than what happens in the field situation. So in the field, we wanted to look at all different variables changing to see how they influence nitrogen mineralization of the cover crop residue, and therefore how much of the nitrogen is stored in the residue over the rainy season and what would be our risk of leaching.
So just to review, our field trial had 3 main treatments. On the left here, we had a fall-terminated Merced rye cover crop that was planted mid-August and terminated mid-October. So it had about 60 days of growth. And those residues were incorporated in beds over the winter, so there was no roots growing. There was no cover crop growing over the winter, just the residues. In the center, we had an overwintered rye cover crop treatment that was planted mid-October and terminated at the beginning of March. And then lastly, on the right, we had a no cover crop treatment that was left in fallow beds and kept weed-free from mid-August until the planting of the spring lettuce crop in the spring. So here we're going to start with cover crop nitrogen uptake. So we found that in the fall terminated cover crop that was grown around 60 days, that cover crop took up just as much nitrogen as the winter cover crops in about half the time, because those winter cover crops grew for around 120 days over the winter. And this is really promising for fall terminated cover crops, showing that they have the ability to accumulate as much nitrogen in their biomass as winter cover crops, usually around 75 to 100 kilograms per hectare in half the time. And so one goal of this study was to track the changes of the C to N ratio for the fall terminated versus the winter cover crops. And as you can see on this plot here, both cover crops produced shoot residue that exceeded a C to N of 20:1. And it's important to grow the cover crop for long enough to maximize nitrogen uptake and then to dilute the nitrogen with carbon, which bumps up that C to N ratio. because this would slow the nitrogen release once the residue is incorporated into the soil. The shoot residue of both cover crops reach a C to N of 34:1 by the time they were terminated. So the green is the fall terminated and the red is the winter. While the incubation study showed reduced nitrogen mineralization of the residue, this mainly occurred at high C to N ratios. And it's important to note that in many cases, the C to N ratios of fall terminated cover crops in on-farm plantings in the Salinas Valley seldom reach or exceed a C to N of 20:1. And thus it's much lower than what we found in our study.
So our third set of data is the soil pore water data. We collected samples at 90 centimeter depth using suction lysimeters. The plot shows on the y-axis the number of samples that were collected for fall-terminated cover crops on the left and the fallow on the right. The darker the blue, the cleaner the water sample compared to the orange, which represents the water samples with nitrate contamination concentrations up to or between 30 and 50 ppm nitrate nitrogen. So all 7 water samples in the across the main 4 rain events that happened last year, we took 7 samples in both treatments, and then in the fallow, all 7 water samples exceeded the drinking water standard for nitrate nitrogen of 10 ppm, whereas in the fall terminated cover crop, only 3 of the 7 samples exceeded this drinking water standard. So our fourth and final set of data is now our soil nitrate data. So also from 60 to 90 centimeters, so the deepest level of the soil that we measured. And this is consistent with our water samples. So this box plot shows all the nitrate nitrogen concentrations from August to May, so the complete duration of the field study, and again, from the deepest soil measured. So the y-axis is nitrate nitrogen, and the x-axis are 3 treatments, where green is the fall terminated cover crop, blue is the fallow or no cover crop, and red is the winter cover crop. The lowercase a, b, and c above each treatment signify the statistical numbers and show that those treatments had different concentrations. So fallow has the highest soil nitrate, fall cover crops intermediate, and winter cover crops are the lowest. The black horizontal line that you see is again at 10 ppm, and we know that this is soil data, not necessarily what would be in the water itself, but it gives you a reference point. So as you can see, there were only 1 sample above this line for the winter cover crops compared to many samples for the fallow and a few for the fall terminated treatment. Year 2, which is what this data, the last 4 plots we've shown you, has come from, was really a best case scenario for a field trial where rainfall was below average, and we were able to keep our fallow plots weed free using a flame weeder instead of a cultivator, which reduced disturbance. In this year of our experiment, we found that the fall terminated cover crops did have lower nitrate nitrogen than the fallow at depth. However, now we'll show you 2 additional fields that we also took soil samples from. And here's the small plot that I just showed you from the last slide, with the fall cover crops having lower soil nitrate than the fallow. Now we added a second plot. This is the first year of our organic field trial that Sierra earlier described, and we found that the fall terminated cover crop plots actually had a higher nitrate at depth than the fallow, as you can see by the different lowercase letters above the plots, indicating the statistical differences. And third, this is a plot from a 1-year conventional trial we did also in the Salinas Valley. And we found that fall terminated cover crops and the fallow plots didn't have statistically different soil nitrate concentrations at depth. Again, the axis of all 3 plots have different scales, indicating different concentration ranges of nitrate. And as a reminder, the black horizontal lines are a reference, so you can see for the 3 fields. So to really reiterate our point, all 3 fields have different results for the soil 60 to 90 centimeter nitrate concentrations.
To answer our main question, can these non-legume fall terminated cover crops reduce nitrate leaching? Looking at the 4 plots that we just showed you for the second year of our field trial that showed nitrogen uptake, C to N ratio, soil and pore water nitrogen concentrations, both at depth, we can conclude that in some cases, yes, but we want to be really cautious for a number of reasons. Firstly, we only have 1 year of field data from the second year of our field trial that supports this. Our first year had many challenges with winter management of the winter fallow beds. And so for simplicity, we didn't get into that data. Additionally, the nitrogen loads of some conventional farm soils that Anna just showed, we measured 10 times higher nitrate concentrations than the organic study that we did. And so those high nitrogen levels would greatly exceed what a cover crop could potentially take up. Secondly, the C to N ratios of fall-terminated cover crops in our organic field study were much higher, like I said, around 34 to 1 than typical levels on farms, usually less than 20 to 1. With fall-terminated cover crops, the high C to N ratios are really, really important to show slow nitrogen mineralization during the winter. And it's unknown how consistently we can get the fall-terminated C to N ratio up that high. And lastly, while incubation studies help us understand the nitrogen mineralization dynamics, they might underestimate mineralization rates in a real-world field condition because you have fall and winter tillage that would likely increase mineralization rates under variable moisture. So in summary, while some of our data indicates that fall-terminated cover crops can reduce nitrate leaching, we believe that more research is needed to better understand the nitrogen dynamics of this cover cropping research. And with that, we will conclude our presentation, and we look forward to answering any questions.